So in our today lecture, we will discuss in detail about the QTL mapping, quantitative trait locus mapping. The traits which are governed by many genes are known as quantitative traits. So the genes responsible for these traits, they are identified with the help of QTL mapping technique. In QTL mapping, first we will record the phenotypic data, the trait data, the trait whose genes we are interested to determine. So this phenotypic data will be the trait data. Trait data. For example, we are interested to determine the genes involved in plant height in wheat. Plant height in wheat. This is our trait of interest. So we want to determine the genes involved in the expression of plant height in wheat. So for the QTL mapping procedure, first of all, we have to develop a mapping population. A mapping population. Generally, we use the mapping population. The mapping populations which are in general use, these are the F2 population. F2. F2 population can be used as a mapping population, or the recombinant inbred lines, rails, they can be used as the mapping population. Rails, recombinant inbred lines, these can also be used as a mapping populations, and so on. So these are the some of the examples of the mapping population which can be used. So in our present example, we will use the F2 population as a mapping population. So what is the procedure to develop the mapping population? We will cross two inbred lines. Two inbred lines with the contrasting traits are crossed. So for the development of the F2 generation, two inbred lines with the contrasting traits are crossed. For example, in our present example, we will cross two wheat cultivars. The individual, the plants are two wheat cultivars which have tall plant height, and the second one with dwarf plant height. So these are two inbred lines with the contrasting traits. One is with the tall height and the other one is with the dwarf plant height. These two plants, they are crossed, and by crossing these plants we get the F1 generation. So F1 generation will be heterozygous but homogeneous. Heterozygous, it will have at all the loci the two alleles Two forms of the alleles will be present at every locus, but all the plants they will have the same morphology, homogeneous. Then, when one of the ever plants are selfed by selfing the F1, we get the F2 generation. By selfing of F1, we get the F2 generation. This F2 generation is the segregating generation. It will have the maximum segregation. So this F2 population will be phenotyped at the maturity stage. We will get the data of all plants of the F2 generation, record the plant height of all the plants of the F2 generation, mostly the size of the F2 population. It is about the optimum size of the F2 population for the mapping population is 250 individuals. So the data of 250 individuals or 250 plants of the F2 generation is recorded. So this will give the phenotypic data. Then in the next step, we will extract the DNA from these 250 plants of the F2 generation. And then we will select a suitable DNA marker system. So in this case, we will use the SSR markers, DNA marker system. SSRs. These are the simple sequence repeats. So these 250 individuals, they are genotyped with these SSR marker system, simple sequence repeat marker system. The optimum number of the markers to be used must be large enough. So in this case, we will use about 250 polymorphic SSR markers. 200 polymorphic SSR markers. So how we will get these 250 SSR markers? First, the inbred lines, two inbred lines, the two parents to develop the F2 population. These two parents, they are screened for polymorphism with a large number of SSR markers, let's say about 1,000. So out of these 1,000, if we get 250, those are the polymorphic. Polymorphic SSR markers means these 250 SSR markers, they have different forms in these two parental inbred lines. So maybe the size of every each of these 250 polymorphic SS markers is different in these two parental inbred lines, the inbred lines used to develop the F2 population. So then the 250 plants, 250 individuals of the F2 population, They are genotyped with these 250 polymorphic SSR markers and data is recorded. The data can be recorded in two forms, either on the basis of presence or absence of the marker, presence or absence of the band for a specific SSR marker, or another way to record the data of the marker is the on the basis of the size, on the basis of the size of the band in base pairs. In base pairs. So the data can be recorded, the band, the SS, DNA markers, data can be recorded either on the basis of the presence or absence of the band for a specific SS marker, or it can be recorded on the basis of the size of the band in base pairs. So in this way, the 250 F2 individuals, they are amplified with the 250 polymorphic SSM markers, and then these the amplification products, they are visualized by using the electrophoresis technique and then by developing the staining the gel. So the procedure involved in the genotyping process steps. In the genotyping, what are the steps involved? So these can be summarized as number one is PCR procedure, or we can simply say the amplification of the DNA markers in each of the F2 individual. So for this PCR procedure, first we have to extract the DNA from these 250 F2 individuals. The DNA from these 250 F2 individuals is extracted, and then the PCR procedure is taken out by using the PCR mixture and putting the PCR mixture in the thermal cycler. So first is the PCR procedure, then is second is the electrophoresis or the PCR products of the PCR products. Then the obtained PCR products, they are visualized, they are separated on the basis of their sizes by a procedure known as electrophoresis, which is simply a procedure in which a gel is used for loading the PCR products, and then after loading the PCR products in appropriate gel, electric voltage is applied, and against this electric voltage, the PCR products, they move according to their sizes. The If the size of the PCR product is less, it will move with the greater speed compared to the PCR product whose size is more compared to the other PCR products. So after the an appropriate time, This electrophoresis procedure is stopped and then the gel is scanned to visualize the position of the PCR products. Staining of the gel to visualize the bands of the PCR products. So the gel is stained and this staining uh, make the bands of the PCR products visible under a fluorescent light. So the bands they can be recorded against a DNA ladder. So this size ladder can be compared to record the sizes of the DNA band. So in this way, the data, the genotype data is recorded. Recording of band sizes or presence, absence of the bands. So in either form, the data can be recorded either on the basis of the band sizes or the presence or absence of the band. So this data will be the genotype data. So this recording of the band sizes, it will generate the genotype data. So in this way, at the by the application of these procedures, we can record the genotype data for the 250 F2 individuals and for total number of 250 polymorphic SSR markers. This will be the genotype data. And at the maturity stage, the plant height of each of the 250 F2 individuals will be recorded. So this uh, recording of the plant height at the maturity stage for the 250 F2 individuals, this will generate the phenotypic data. The phenotypic data, the trait data, and in this case, the trait we are interested in is plant height. So after these two forms of the data, the phenotypic data and the genotypic data generation, these two types of the data, they are analyzed by computer-based softwares. So the softwares which are used for the different purposes in the QTL mapping, these softwares are computer-based softwares for analysis, for molecular analysis. For molecular analysis of the phenotypic and genotypic data, the software commonly used, the softwares are number one, join map software, join map software. So this join map software or another software which is also used for the same purpose is the map picker, map picker or power marker. So these different three types of the softwares. These softwares can be used to construct linkage map. So these softwares are used to construct linkage map with the help of the genotype data. So we have used 250 SSR marker with the help of joint map or the map maker or power marker. We can construct the linkage map, and this linkage map will show that which marker is located on a specific linkage group or on a specific chromosome, on a specific bead chromosome. So the 250 SSR markers, polymorphic markers, their data is used to construct the linkage map, and this map will show that which particular group of SSR markers they are located together on a specific chromosome. Then after the construction of the linkage map, the QTL cartographer software is used to assess marker trade associations. Assess marker trade associations. So the QTL cartographer software used to assess market trade associations. This analysis tells us which SSR marker is associated with the plant height. Out of 250 polymorphic SSR markers, maybe out of these 250 polymorphic SSR markers, they out of these 250, maybe 220, they are located on the linkage map, and out of these 220, maybe which marker they are associated with plant height. The QTL cartographer analysis will tell us which essential marker is associated with the plant height. It means that which essential marker is closer to the gene responsible for plant height in wheat. So in this way, we can go closer to the gene responsible for plant height in wheat, the genes which are involved in the expression of plant height in wheat. So this is the total procedure which by using this procedure, we can identify the genes involved in a quantitative trait. A trait governed by many genes, and these many genes they can be identified with the help of a QTL mapping procedure. And in this QTL mapping procedure, we will use the phenotypic data, the trait whose genes want to identify, and then for the molecular analysis we also need another data which is known as the genotypic data. To generate the genotypic data we use a suitable marker system, and in this example we have used the SSR marker system, simple sequence repeats. Where these are the DNA sequences which are widely distributed in the genome of the organism. In the genome of every organism, there are certain repeats which are present throughout the genome. These are known as simple sequence repeats. So these, in this case, we have used 250 SSR markers and 250 F2 individuals. We report the genotypic data for the 250 SSR markers by using 250 F2 individuals, and then by using the joint map software or the map maker software or power marker software, we will construct the linkage map out of these 250 polymorphic SSR markers. and maybe out of the 250, about 220 markers, they are located on specific linkage groups. They are present in the pharma group, and this group will show that which group of the markers they are present as a whole on a specific chromosome. Then by using the QTL cartographer software, we will determine the marker trade associations. It means that this QTL cartographer analysis will show us that which essential marker is closer to the gene controlling plant height in the wheat. So we have covered in detail the QTL mapping procedure by taking the example of plant height in wheat.